So, good morning, everybody. I think we are about to be ready to start. It's 3 minutes past the hour, so we still need some people to dial in, but let's get started anyway. Good morning, everybody. Welcome to have you here. Great that you are participating in the third webinar for Nuvisan. My name is Arnold Huisman. I'm the Chief Strategy Officer here at Nuvisan. And today we are in Berlin at the Innovation Campus, very exciting. And when I joined Nuvisan a couple of months ago, I must admit I was quite impressed with one of the departments that we actually are going to be zooming in today, the cross-indication research and their ability to increase the value of your compound. So it's great to have all the people here. So our scientists today, specializing in the cross-indication research, are Charlotte and Eki. They will guide us through the setup for today. So let's not wait any further, and let's get started with Charlotte and their introduction. Please go ahead, Charlotte.
Yes.
Sorry. Thank you, Arnaud, for your introduction, and to all the participants, thank you for joining today's webcast. And it's my pleasure to introduce now Eki to you. So Eki, my head of cross-indication research within the Innovation Campus Berlin.
Yeah, and this is Charlotte. Charlotte is head of our therapeutic research department. So some people outside call it pharmacology. And yeah, we will guide you through what we can offer to you.
Thank you. And with this, I would like to announce first something. So your device is not broken. So this is a glass frame due to known reasons. So it's not a scratch in your device. And the other thing is your microphones are muted by default. So please, in case you have any questions, write them down. We will have a question and answer round at the end of today's webcast. With this, I would like to start. So we will first give you a brief overview about the services which you can have with us at the Innovation Campus in Berlin, followed by the value of cross-indication research by 3 different examples. And of course, at the end, as mentioned before, the live question and answer session. So please write down again your questions and provide them to us afterwards. So this I already mentioned, and afterwards you will be contacted. We have your contact details for the follow-up on today's webcast. So before we start with the content, we have a first live poll. It would be great if you could kindly ask the question you can see now.
Interesting.
Yes. So with this, yes, we have quite a lot of people participating the poll. So thank you very much. So let me know once it's final. It's final? No? No. But we have a clear trend. A very clear trend.
Definitely.
And it shows us that it's worse that we have today's webcast. Sure, you're eager to know.
I think the profile isn't changing much.
Yeah. Okay.
No, so we have somehow like Gauss proportion, so with some people who have worked with Eki and me before, although not with Nuvisan before. And yeah, most attendance of the peak is definitely, no, I never worked with Nuvisan before. So I hope that this can change in future and that we convince you today with our webcast that it's worth to do some work together with us. And with this, I would like to come to the details. So Nuvisan is now a fully integrated CRO, which can offer you services starting from basic research until early clinical trials. You have here the chance, no matter whether you're a pharma company, a biotech company, venture capital company, or an academic institution, to get those services from us all out of one hand, which we think is a quite nice and rather unique opportunity you have. And how does it come that Nuvisan now offers the services starting from basic research? This is due to the latest acquisition of Nuvisan, namely the transfer of a fully integrated research unit from Bayer in July this year to the Nuvisan Group. So we were part of this transfer, and the other departments which we're in is lead discovery Berlin, then life science chemistry, and DMPK and toxicology services. So really offering services from the target ID until IND preparating studies. And today we will focus on therapeutic research. And here I would like to show you what we can offer in these integrated packages. So we have quite a lot of activities ongoing, from expression profiling until genetic manipulation, compound target, as well as patient segment characterization. As said, we do have a proof track record, which is followable by several publications, and all the red underlined authors on these publications are now part of the ICB. So really showing you that with our experience we have from our former times, we contributed to the development and discovery of several compounds in the past. I would like to highlight those activities which therapeutic research can offer by one package performed, which was a project-tailored in vivo characterization and biomarker identification, in this case for an ADC. So on the left-hand side, you can now see in subcutaneous ovarian cancer models and the tumor activity inside the model. But next to the subcutaneous tumor model, we of course performed also an orthotopic ovarian cancer model. And furthermore, we had a question from the clinicians in preparing the phase 1. Their question was, what is the cutoff? How many tumor cells need to be actually positive within a tumor that a patient has a likelihood to benefit from the treatment? And in order to answer this question, we had generated stably transfected cell lines, mixed them so that we had tumors which growed with a different density of positive cells, subsequently treated those tumors. And in the lower panel, you can see that in most of the conditions, the tumors were shrinking. So the bars showing to the minus are shrinking tumors over time, showing us that the number of positive tumors was a rather not that important factor as thought before. And here demonstrating a bystander effect of our compound on the tumors, which was very nice for the design of the clinical trials. And with it, we are too early with the question. I go to the next slide and we come back? So sorry for the mistake. So this is rather the normal way how you perform your drug discovery campaigns. You have a target idea, you have a disease of interest, and then you are identifying and characterizing the compound as well as of the disease of interest. And during this value chain, you have quite a lot of compounds in your hand. So you have a late research compound, which is already active in different tumor models. You have a development compound, which already entered clinical trials. You might have shelved assays, which were on hold for different reasons. And you have marketed drugs in your pipeline. And one thing which you can do is that not only development of those compounds in your interest, disease of interest, but to use reverse pharmacology in order to identify additional segments in which you can work. So here we can offer with the cross-indication research a process which allows the drug repurposing, life cycle management of your compounds, and also the de-risking of your clinical development. And this is done by a mode of action-driven characterization of diseases and therefore diversification of indication space, and leading to an increased probability of clinical success, also to new or additional IP, and also to an increased probability of return on investment. And this all because you have finally more errors in your query. And with this, we are now coming to the poll. Which I again would like to ask you to answer.
All right. We see something.
Thank you for participating.
Yeah. Again, interesting. All right. Is it free? Mm-hmm. Okay. So you see, this poll is a little bit different. So it's fairly balanced, actually. So we see many people with late research compounds, but also development companies, marketed drug shelf assay, very interesting. So I'm really looking forward to guide you now through the further slides. I think there will be something interesting for all of you in it. So thanks, Charlotte, for providing the frame. And now I will dive a little bit deeper into the cross-indication project or program. So again, this is the value chain that you've seen many times, and this is a little bit more generic. So the one you probably are familiar with, from basic research to market, and all the assets that come out of these different phases. And clearly all these phases require investment to get through it. That's obvious. And it's also clear to all of you that this investment is relatively little in the early phases. So research is not so expensive, but still costs are accumulating. And at the end of a preclinical program, you still might have costs of 10 to 30 millions accumulated, depending also if you are counting different project number, but it's relatively minor as compared to the costs you get later in phase 2. So here you have to deal with 20 to 40 millions to run a phase 2 or a POC study. And the point here is that if you look on the probability of success, or inversely the attrition rate in the different phases, you also have to see a particular situation in phase 2. So here, the whole industry faces the problem that there is a very high attrition rate in phase 2. So 2/3, 70% of the project are failing, mainly due to efficacy. So that's the main reason. And if you see that, then you obviously have to also see the possible reward you can get if you make that hurdle, if you get over it. And that is a very dramatic increase if you compare the numbers or the amount of money you can get, the value of your asset, preclinical asset, at the end of the preclinical profiling phase. So if you get 10 millions for a late preclinical project, then you may be happy if you get the costs out of it. But if you are able to get such a project through phase 1 and then into phase 2 or into POC, it doesn't necessarily have to be a phase 2. It can also be a proof of mechanism, but something that works in humans. So if you make that proof, then you have really elevated the value of your asset. And for us, and for probably the whole industry, if it's biotech or big pharma, the question is how to get there. I mean, to get through this high attrition rate, or inversely,